History of Life on Earth. Chapter 2, First Life and the Age of Prokaryotes. The 2 billion years when nothing had a body. 2.1 The Invisible Empire. Set yourself down on the shore of an ocean 3 billion years ago. The sun is up, but it looks wrong, smaller, paler. Its light filtering through a hazy orange sky that holds no oxygen you could use. The continents are strange stubs, small and unfamiliar. There is no grass, no moss, no lichen, no scum of green on the rocks. The land is bare stone and sand, sterile as the surface of Mars. There is no sound but the wind and the surf, because nothing on this planet can make a sound. Nothing moves. Nothing swims. Nothing flies. You would be standing on a living world and you would see almost nothing alive. Look closer though. In the warm shallows, the rocks are slick with a film, a slime, faintly purple, pink, and deep rust brown, layered and quilted. It is not much to look at. It is also, at this moment, the most sophisticated thing in the known universe. A city of billions, running chemistry no laboratory could match, and busy, slowly, patiently, over spans of time that make our whole species look like a spark, remaking the entire planet. This chapter is about those 2 billion years. Nearly half of Earth's history passed with prokaryotes, single cells with no nucleus, invisible to the naked eye, as the only life there was. They were not waiting around for something better to come along. In that span, they invented nearly every metabolic strategy life still uses, rewrote the chemistry of the ocean and the sky, precipitated most of the world's iron out of the sea, froze the planet, and triggered what may have been the greatest mass extinction that has ever occurred. Everything that came afterward, the trilobite, the fern, the dinosaur, is a late elaboration on the foundation they laid. 2.2 The Ancestor of Everything. Start with something odd. An oak tree, a hammerhead shark, a slime mold, a bacterium boiling in a Yellowstone hot spring. And the person reading the sentence do not appear to have much in common. But go down to the molecular level and the differences evaporate. All of them store information in DNA. All of them use very nearly the same genetic code, the same 3-letter words meaning the same amino acids, a dictionary shared across every kingdom of life. All of them build proteins on ribosomes made of RNA and protein. All of them spend energy in the currency of ATP. All of them are wrapped in a lipid membrane. That is not the kind of thing that happens twice. Anything could have worked. There is nothing sacred about this particular code, this particular currency. That every living thing on Earth uses the same arbitrary conventions means all living things inherited them from a single ancestral population that we call the last universal common ancestor, or LUCA. Now, 3 corrections, because LUCA is almost always misunderstood. LUCA was not the first living thing. Life had already been running for a long time, evolving, competing, failing, before LUCA came along. LUCA was not an individual. It was a population, a fuzzy, interbreeding, gene-swapping crowd of cells, not a single lonely Eve floating in the dark. And LUCA was not simple. By comparing the genes shared by nearly all bacteria and all Archaea alive today, researchers can back-calculate what their common ancestor must have carried. The reconstruction is startling. LUCA operated right at the boundary of modern genetics. While it may still have relied on a more primitive RNA genome, leaving bacteria and Archaea to later invent standard DNA replication independently, it already transcribed genetic codes into RNA and translated them into protein on ribosomes. It had dozens of core metabolic enzymes. And it generated its energy by pumping protons across its membrane and letting them flood back through a molecular turbine. In other words, LUCA was already a working, competent, thoroughly modern cell. Everything hard had already been solved. There is one more thing worth pausing on, because it is the strangest fact about LUCA and the easiest to miss. LUCA is not special because it was the best. LUCA is special because it was lucky. There were almost certainly other lineages alive at the same time, cousins, rivals, other experiments in being alive. Every one of them died out and left nothing behind. LUCA is simply the most recent ancestor whose children are still here. LUCA is not the ancestor of life. LUCA is the ancestor of the survivors. And where did the survivor live? The genomic evidence points to a cell that liked heat, could not tolerate oxygen, and made its living by combining hydrogen and carbon dioxide. Many of its most ancient enzymes are built around little clusters of iron and sulfur. Its power system was a proton gradient across a membrane. Hydrogen, carbon dioxide, iron and sulfur. A proton gradient. Read that list again and then look back at Chapter 1's alkaline hydrothermal vents, which offer exactly those four things and nothing else. To entirely separate lines of evidence, the geology of the seafloor and the genes inside modern microbes are pointing at the same address. 2.3 The continent nobody had noticed. In the early 1970s, a quiet, difficult, obsessive microbiologist named Carl Woese set out to do something nobody had managed: build a family tree of the microbial world. The problem had always been that microbes have no anatomy to compare. A biologist can tell a bat from a bird by the bones. But under a microscope, one rod-shaped bacterium looks much like another, and appearances tell you nothing about ancestry. Woese's insight was to stop looking at organisms and start reading molecules. He chose ribosomal RNA, a component of the ribosome, present in every living thing without exception, and slow to change. It was the one yardstick that could be laid against everything. The work was brutal. He labeled the RNA with radioactive phosphorus, chopped it into fragments, spread the fragments across a film, and read the resulting smears of dots by eye, in a darkened room, hunched over a light box for years. In 1977, the dots said something impossible. A group of methane-producing microbes, creatures everyone had filed under bacteria without a second thought, turned out, at the molecular level, not to be bacteria at all. Their ribosomal RNA was as different from a bacterium's as a bacterium's is from yours. They were not a branch of the bacterial tree. They were a separate tree. Life, it turned out, was not divided into two great groups but three: bacteria, Archaea, and Eukarya, the domain that includes plants, animals, and fungi. See Chapter 3. Woese had found an entire continent of life hiding in plain sight, in hot springs and swamp mud and the stomachs of cows, and nobody had ever noticed. Because it looked like everything else. The reaction was not warm. A great many senior scientists thought he was wrong, and said so. The evidence kept accumulating anyway. By 1990 the three-domain system was formalized, and today it is the map on which all of biology is drawn. What actually makes an Archean different? Not size, not shape, and not the absence of a nucleus. They have none, and neither do bacteria. The differences are chemical, and they run deeper than any visible feature could. Bacteria build their cell walls from a mesh called peptidoglycan. Archaea never do. Archaea have a large, elaborate machinery for reading DNA that resembles ours far more than a bacterium's. One of the first hints that we are closer cousins to the Archaea than either of us is to bacteria. But the deepest difference is the membrane. And here is where it gets remarkable. Bacteria, and you, build membranes out of straight fatty acid chains attached to a glycerol backbone by one kind of chemical bond. Archaea use branch chains, attached by a different kind of bond, and, most tellingly, attached to a glycerol backbone that is the mirror image of yours. Think back to Chapter 1 and the puzzle of handedness. This isn't an exception to that rule. The fundamental pieces remain the same, but it is a profound structural fork in the road. When constructing their cellular scaffolding, the two domains use entirely different enzymes to tag their central glycerol molecules. The result is that their membrane backbones are constructed as exact, non-interchangeable mirror images of one another. Nobody is entirely sure what that means. It is called the lipid divide, and one intriguing possibility is that LUCA did not have a modern membrane at all, that it lived inside the mineral pores of a vent, using the rock itself as its wall, and that the two lineages only built proper membranes for themselves after they split, each one picking a hand at random, which would mean that when life finally walked out of the rock, it walked out twice. One correction: while we're here, Archaea are often described as extremophiles, lovers of boiling acid and saturated salt. Many are, but many are not. They drift by the billions in the open ocean, live in ordinary soil, and inhabit your intestines right now. They are not exotic. They are simply quiet, and we were slow to notice them. 2.4 What the rocks remember. We have no photographs of the first 2 billion years, and very nearly no fossils in the ordinary sense. What we have are three kinds of whisper. The mats. In the ancient rocks of the Pilbara, in Western Australia, There are structures about 3.5 billion years old that look like stacks of stone, pancakes, or domes built up from fine, wavy layers. They are called stromatolites. And they are not rocks that happen to be layered. They are rocks that were built. A mat of microbes grows on the seafloor. Sediment settles and buries it. The microbes climb up through the grit toward the light and grow a new mat on top. More sediment settles. Repeat for 1,000 years and you have a dome. The best part is that you can still go and see them alive. In the hot, hypersaline shallows of Shark Bay, on the same Australian coast, living stromatolites are still being built by microbial mats today, exactly as they were 3.5 billion years ago. You can wade out into knee-deep water and stand among them, and for a moment you are standing in the Archean. The chemistry. Life is a picky eater at the atomic scale. Carbon comes in two stable forms, a lighter one, carbon-12, and a heavier one, carbon-13, and the enzymes that pull carbon out of the environment work fractionally faster on the lighter atoms. So living matter always ends up slightly enriched in carbon-12 relative to the world around it. This is a fingerprint, and it survives even when the organism doesn't. Burn the cell away, compress it into a smear of graphite, bury it for 4 billion years, and the isotope ratio still whispers something alive made me. Rocks from Isotopic, in Greenland, dating to about 3.7 or 3.8 billion years ago, carry carbon with exactly that lightness. If it is what it appears to be, it is the oldest trace of life on Earth, pushing the origin of life almost uncomfortably close to the end of the era, when the sky was still full of falling asteroids. It is also contested, because non-living processes can occasionally mimic the same signature. The argument is not settled. The bodies, structures interpreted as fossilized cells turn up by around 3.4 billion years ago in Australia and South Africa: tiny dark threads and spheres in ancient chert. Here the difficulty is almost philosophical. At that scale, a fossilized bacterium and a purely mineral artifact, a bubble, a crystal, a wisp of carbon squeezed between grains, can look maddeningly alike. Several famous claims of the oldest fossil have been challenged and rechallenged for decades. That's worth sitting with, actually. This is what real science looks like at the edge of what evidence can support. Not a clean answer, but a long, careful, unglamorous argument about whether a smudge is a cell. The whispers are faint, but they all say the same thing, and they say it early, by 3.5 billion years ago. The planet was already inhabited. 2.5 eating rocks. Now, a question that sounds simple and isn't. What did life eat before it invented photosynthesis? Long before the first cell figured out how to harvest sunlight, organisms had to rely on the raw chemistry of the planet. Hydrogen seeping from the crust, carbon dioxide dissolved in the sea, sulfur compounds bubbling from vents, iron in the water. And so the first organisms ate rocks. Or, more precisely, they ate what rocks and gases could give them. The technical name is chemolatotrophy, and to understand it you need one idea, which is the single most useful idea in all of bioenergetics. All of life is electron traffic. Electrons can sit at high energy, or low energy, and they naturally fall from high to low, the way water falls downhill. Left alone, that fall releases its energy as useless heat. But if you can route the fall through a machine, a water wheel, you can tax it on the way down. That's all an organism is. Every living thing on Earth, from the microbe in the vent to the reader of this sentence, is a device for finding electrons at high energy, escorting them down to low energy, and skimming a profit from the descent. The early cells found their electrons wherever the geology offered them. Some ran methanogenesis, combining carbon dioxide with hydrogen, releasing methane as waste, needing no oxygen and no light. Archaea still do this in swamps and rice paddies and the guts of cattle. The methane in Earth's atmosphere, today, is largely microbe exhaust. Others ran their electrons down onto sulfur compounds. Others survive by fermentation, cannibalizing small, organic molecules, and pocketing a tiny profit, a low-yield, ancient, no-frills strategy that is still running in every batch of beer and every loaf of rising bread. And underneath all of these strategies, without exception, is the same machine. The cell uses its captured energy to shove protons across its membrane, pumping them out, piling them up on the far side. Now there's a difference in pressure, an imbalance, a charged battery. And built into the membrane is a channel the protons can rush back through, a squat protein turbine called ATP synthase, which the flooding protons physically spin, like water spinning a mill wheel. A real rotation, a real axle, perhaps 100 turns a second, and every rotation stamps out ATP. You have somewhere around 10 million billion of these turbines in your body, spinning right now, in the membranes of your mitochondria. When a biochemist named Peter Mitchell proposed this in 1961, that cells make energy not through some tidy chemical intermediate, but by pumping protons across a membrane, and letting them fall back through a turbine, the field found it faintly ridiculous. Mitchell was in poor health. He left academia, restored a manor house in rural Cornwall, and set up his own laboratory there, largely funding it himself. He kept working. In 1978 the Nobel Committee gave him the prize in chemistry, and today his mechanism is understood to be as universal as the genetic code. Which brings us back to the vents. Chapter 1 described the natural proton gradients seeping through the mineral pores of an alkaline hydrothermal chimney, a battery running for free, before anything was alive to use it. The gradients inside your cells are not nearly similar to that. They are the same trick, in the same direction, made portable. Life did not leave the vent behind. It packed it up and took it along. Every cell you own is a hydrothermal vent, wearing a membrane. 2.6 The invention of eating light. Somewhere in the deep Archean, a lineage of bacteria did something no chemistry on this planet had ever done. It stopped waiting for the Earth to hand it high-energy electrons, and started making them, by catching a photon of sunlight and using its energy to kick an electron uphill. The first version, an oxygenic photosynthesis, is still with us. Purple and green sulfur bacteria run it today in stagnant lakes and sulfurous muck. Light hits a pigment, an electron is boosted to high energy, and the cell rides it downhill through a chain of carriers, pumping protons all the way. But the electron has to come from somewhere, and this early version stole it from hydrogen sulfide, the rotten egg gas of volcanoes and vents. The waste product was not oxygen. It was elemental sulfur. Some of these bacteria still stockpile it as glittering yellow granules inside themselves, like tiny hoards of treasure. It worked, but it had a hard ceiling. Sulfide is a local resource. It bubbles up here and not there. The bacteria could only ever live where the geology fed them. Meanwhile, all around them, in unlimited supply, sat the most abundant electron-bearing molecule on the surface of the Earth: water. The trouble is that water does not want to give up its electrons. It is an extraordinarily stable molecule. That's why the oceans are still here, and prying it apart takes vastly more energy than one photosystem could ever deliver. The solution, when it finally evolved, is one of the great pieces of biochemical engineering in the history of the planet: 2 photosystems, wired in series. A I-stage rocket. The first photosystem catches a photon and boosts an electron. The electron runs downhill through a chain, doing useful work, and then, at the bottom, exhausted, it is caught by a second photosystem and boosted a second time by a second photon, up to an energy high enough to do the truly hard chemistry. The 2 systems most likely arose from the duplication of a single ancestral one, followed by long specialization, evolution's favorite move, copying a tool, and then reshaping the copy. And at the business end of this contraption sits the machine that tears the water apart. It is astonishingly small, a cluster of 4 manganese atoms, and 1 calcium, bound in a lattice with oxygen, structured like a microscopic fragment of mineral, held perfectly in the grip of a protein. It seizes water molecules, strips away their electrons, and their hydrogen, and throws away what's left. What's left is oxygen. Take a breath. Essentially every oxygen atom that just entered your lungs was pried off a water molecule by a speck of manganese inside a bacterium or inside a plant. There is no other significant source. The air you are breathing right now is, in the most literal sense, waste, the discarded byproduct of a chemical reaction that microbes invented to solve a supply chain problem. The inventors were the Cyanobacteria, and as far as anyone can tell, they invented it exactly once. Every blade of grass, every leaf on every tree, every strand of kelp, and every green cell in the ocean is running oxygenic photosynthesis on machinery inherited through a long and startling chain of events we'll come to in Chapter 3, from Cyanobacteria. One lineage, one time, several billion years ago, and the entire green world descends from it. When that's genuinely disputed, an oxygenic photosynthesis was probably running by 3.4 billion years ago. Oxygen-producing photosynthesis came later. But how much later? Hundreds of millions of years before the atmosphere changed, or only shortly before, is one of the liveliest arguments in the field. What isn't disputed is what happened next. 2.7 The Great Poisoning. Here is the thing about oxygen that we, its beneficiaries, tend to forget. It is a molecular arsonist. O2 is aggressive, unstable in the chemical sense, ravenous for electrons. It rips them out of whatever it touches. It shatters DNA, unfolds proteins, and turns the fats in a membrane rancid to an organism that evolved in a world without it. Air is fire, and the cyanobacteria were making it, and making it, and making it for a long, long time before anything happened at all. Because at first, the planet simply drank it. The Archean ocean was loaded with dissolved iron, which cannot exist in that state today, but can in the absence of oxygen. As the first free oxygen appeared, the iron grabbed it. The sea rusted. Iron oxide, insoluble, snowed down onto the seafloor and settled into layers. When the microbes bloomed, the water rusted and a layer of iron oxide settled. When the bloom failed, a layer of silica settled instead. Bloom, rust, pause, silt, bloom, rust, over and over, for hundreds of millions of years, until the seabed had recorded the whole slow struggle in stripes. We call these banded iron formations, and they are among the most beautiful rocks on Earth. Bands of deep red and silver gray, and black, folded and polished, like frozen weather. They are also where nearly all of the world's iron comes from. Look around the room. If there is steel in the building, girders, screws, the frame of the chair, the body of the car in the driveway, the bridge you crossed this week, that iron was dissolved in a Precambrian sea, rusted out of the water by the exhalations of pond scum, buried, compressed, uplifted into mountains, and finally dug back out by us. Human civilization is built out of microbial pollution. Eventually, though, the sinks filled. The dissolved iron ran out, the reactive rock was consumed, and the oxygen, with nowhere left to go, began to accumulate in the air. That is the Great Oxidation Event, roughly 2.4 to 2.0 billion years ago, and it was a catastrophe on a scale the Earth has rarely matched. The dying, for every organism that had evolved in an oxygen-free world, the new atmosphere was lethal. There is a decent argument that this was the largest mass extinction in the history of the planet, and we will never know, because there is no way to count the dead. No bones, no shells, no bodies. An entire biosphere may have been annihilated, and left us nothing but an absence. The survivors did what survivors do, they hid. They retreated into the places oxygen could not follow: deep mud, waterlogged soil, sealed sediments, hot springs, and eventually the airless interiors of animal guts. Their descendants are still down there, still poisoned by a whiff of air, still living as though the great oxidation never happened. The anaerobes did not all die. Most of them went underground, and never came back up. The freeze, there is a second consequence, and it is stranger. The young sun was significantly fainter than it is today. By rights, the early Earth should have been frozen solid. What kept it warm was a thick blanket of greenhouse gases, and methane was a crucial part of that blanket. But methane and oxygen cannot coexist. Oxygen destroys methane. As O2 rose, the methane fell out of the sky, and the greenhouse blanket, which had been holding a dim sun's warmth against a cold universe, thinned. Around the same time, the Earth appears to have plunged into the Huronian glaciation, one of the most severe ice ages it has ever suffered, with some evidence pointing to ice reaching all the way toward the tropics, a possible snowball Earth. So life rewrote the composition of its own atmosphere, poisoned the surface world, and helped plunge its own planet into a deep freeze. Remember this pattern. It is the first time we will see it, and it will not be the last. The shield, and then, finally, something the catastrophe gave. High in the atmosphere, ultraviolet light began knocking oxygen molecules apart and reassembling them into a three-atom form, ozone. Slowly, over immense spans of time, a thin haze of it gathered in the stratosphere, and began absorbing the ultraviolet radiation that had been sterilizing every exposed surface of the planet. Nothing needed it. Nothing was living on land. Nothing could, but it kept accumulating anyway, patient and pointless. An umbrella held open over an empty room, and it would still be there, waiting, when the first green film crept out of the water nearly 2 billion years later. 2.8 Learning to breathe the poison. Not everything hid. Some lineages, out on the frontier where oxygen was accumulating, did something far more interesting than tolerate it. They found a use for it. Go back to the electron waterfall. The energy a cell can extract depends on how far the electrons fall, and the drop is set by whatever molecule is waiting at the bottom to catch them. Anaerobes were working with poor catchers: sulfur, carbon dioxide, various mediocre acceptors that don't pull very hard. Oxygen pulls ferociously. That's precisely what makes it dangerous, and it is precisely what makes it valuable. Put oxygen at the bottom of the chain and the electrons fall from a much greater height, and the cell can tax the whole descent. The numbers are not subtle. A cell fermenting a molecule of glucose scrapes out about 2 ATP. A cell running aerobic respiration on the same molecule of glucose can extract upwards of 30 plus ATP, more than 15 times the energy of fermentation, harvested from the exact same food. The deadliest molecule on the planet had just become the most profitable one on the planet. The poison became the prize, and that changed what was affordable. Complexity is expensive. Large genomes are expensive. Large cells are expensive. Movement, sensing, internal architecture. All of it costs energy that simply had not existed in the budget before. With aerobic respiration, life suddenly had a much larger income, and could begin, for the first time, to contemplate spending it. There is one more thing to say about these oxygen-breathing bacteria, and it is the hinge on which the rest of this book turns. At some point in some unremarkable stretch of ancient sea, one cell trapped another inside its outer architecture. It may have started as a slow, predatory entanglement, or a metabolic partnership gone deep. Except that this time, the internal cell was an oxygen breather, and this time, for reasons we can only guess at, it was not digested. It stayed, 2.9 the length of the silence. We are very bad at feeling deep time, so let's try compressing it. Squeeze the whole 4.6 billion year history of Earth into a single calendar year. The planet forms on January 1st. The first traces of life appear in the first week of March. Stromatolites are building their domes by the end of March. Cyanobacteria are pumping oxygen into a world that swallows it whole for months. The great oxidation event, the poisoning, the freezing, the rusting of the sea, lands around midsummer, late June. And the first cell with a nucleus, the first cell complicated enough to be called anything more than a bag of chemistry, does not appear until around the second week of August. March to August, more than 5 months of the year, and the most advanced thing on the face of the earth is a single cell without a nucleus. Animals, when they finally arrive in mid-November. Everything you have ever seen with your eyes, every tree, insect, bird, fish, and person, every forest and coral reef, all of human history, compressed to the final few seconds before midnight, is crowded into the last 6 weeks of the year. And in all those quiet months, the simple cells were not idle. Prokaryotes invented fermentation. They invented photosynthesis twice, and oxygenic photosynthesis, which changed the sky. They invented anaerobic respiration, and then aerobic respiration. They invented the rotary flagellar motor, an actual outboard engine with a drive shaft, spun by the very same proton gradient that runs the ATP turbine. They perfected the genetic code, the ribosome, DNA replication, and DNA repair. Everything essential that happens inside you was worked out by microbes before your ancestors were even a possibility. Consider one example, my favorite. Nitrogen makes up 78% of the air, and every protein and every strand of DNA in your body requires it. But atmospheric nitrogen is bound up as in two atoms welded together by one of the strongest bonds in all of chemistry, and it is for practical purposes inert. Almost nothing can touch it. Bacteria can. An enzyme called nitrogenase, built, inevitably, around a cluster of iron, sulfur, and molybdenum, another chip of mineral wrapped in protein, cracks that bond open at ordinary temperature and ordinary pressure, quietly, in the soil, in root nodules, in the sea. When human chemists finally learned to do the same thing in the 20th century, it took a factory, temperatures around 400 degrees Celsius, and pressures of hundreds of atmospheres. Every nitrogen atom in your body was pulled out of the air either by one of those factories, or by a microbe. For most of the history of life, and for most of the nitrogen in the living world, it was a microbe. They also broke the rules of inheritance. Prokaryotes routinely passed genes sideways, swapping them cell to cell, picking up loose DNA from the water, receiving deliveries from viruses. Genes flow across lineages that separated billions of years ago. This is why antibiotic resistance can spread through a hospital in a matter of months, and it is one reason reconstructing the earliest history of life is so maddeningly difficult. Down at the bottom, the tree of life is not really a tree. It is a web. 2.10 The Empire that never ended. It is tempting to read this chapter as a prologue. The primitive stuff, the warm-up act, the long dull wait for the real show to begin. Resist that. Prokaryotes did not lose. They did not give way. They are still, by any measure you choose except sheer physical size, the dominant form of life on this planet. There are something like a nonillion of them, a billion trillion, which is more cells than there are stars in the observable universe, by a wide margin. All the bacteria on Earth outweigh all the animals many times over. They live in boiling water, in solid ice, in brine that would pickle you, in acid that would dissolve you, in reactor coolant, and in solid rock kilometers beneath your feet, eating hydrogen in total darkness, dividing perhaps once a century. They also live in you. You are carrying roughly as many bacterial cells as human ones. You are not an organism with some microbes on board. You are a landscape they inhabit, and much of what your body does, digesting, training your immune system, regulating your own moods in ways still being worked out, it does with their help. So the honest picture is not that simple life was superseded by complex life. It's that complexity is a recent, local, energetically expensive flourish added on top of a microbial world that was already running the planet's chemistry, and has never stopped running it. The atmosphere you breathe, the nitrogen in your proteins, the iron in our cities, the carbon cycle that keeps the whole system in motion, all of it is theirs. We are guests in a house they built and still maintain. But something did change. Around 2 billion years ago, in the wreckage and the opportunity of the Great Oxidation, one cell took another cell inside itself, and did not digest it. The captive stayed. It kept breathing oxygen. It kept making ATP, and it began, slowly, to hand its energy over to its host. Nothing in the history of life had ever produced so much power inside a single cell. And with that power, cells were about to start doing things that had been unaffordable since the beginning of the world. That is Chapter 3,